our presentation. My presentation will be restricted to integrated disease management. And this integrated disease management, we will see what is plant pathology. Generally, we will see what are the different plant pathogens and which are the different diseases, and how the disease we manage with integrated disease management technology, or you can say different methods, which methods includes in integrated disease management. To find out this integrated disease management, we have to see some background of and significance of plant diseases. You know, why this plant pathology branch is emerged in the plant science discipline, because of these diseases. You know, plant diseases reduces the quality of the produce, quality and quantity as well as, and you know, all these pests and diseases cost 30% loss to the adequate produce. Between this due to only diseases, there are 10% loss in food production. And you know, there are so many diseases also entered in our country through exotic mode. Then we have so many diseases entered in our area because of lack of information and lack of restriction. Then each year we found out there is billions of dollars loss is due to diseases worldwide, and this pathogen continually evolve, because continuously we are developing some disease management practices along with the pathogens also evolving themselves with different races and strains, and that's why they are also building resistance against different chemicals which we produce. That's why this is the challenge between pathogen and humans to produce good agricultural produce. You know, this is the great great Irish famine, which was occurred in 1845 and 50s. You know, this is the disease. This is the Irish famine, which is responsible for birth of discipline. And you know, how disease can change the economy of any particular country. This is the great example that, you know, this Irish famine is happened because of the potato disease, it is called as lead blight of potato, and caused by very devastating phytophthora. And this phytophthora fungus is now also is causing so many diseases in various crops, and changing its nature of damage. This great Irish potato famine, you know, in 1845, millions of people are migrated from this Irish country to Great Britain, United States, and Australia, because of there was no food due to infestation of lead blight of potato caused by phytophthora infestations. And this is the severity of that disease, and that's why this country economically disturbed, and so many people migrated, and deaths are around 42%. Therefore, this Anton de Bari in 1861. After that Irish famine, then Anton de Bari then established that diseases are caused by microorganisms called fungi. And after the invention of microscope, then people are understanding that how the diseases are established. Then he established that the phytophthora infestation is the main causal agent and responsible for this potato lead blight. And from that stage, the beginning of plant pathology, branch of plant pathology emerged in plant science subject, and then so many inventions. Then later on, before our just independence in 1943-44, our country also has one decline called as famine, called Bengal famine in India. And here the crop is paddy, and the disease is Helminthosporium blight of paddy. and that disease also causes famine in Bengal, and because that time our primary food is paddy, and due to this blight disease, so many people this caused around 2.1 million people. Weeds causes plant, insect pest causes losses simultaneously. Causes of diseases also causes losses in plant. Around 10% losses are causes due to diseases. And the major food crop of our country is wheat and rice. And major fungal organism which is causing potential threat to our food security in rice. We have blast disease around this rice blast was and which has various host range have 50 classes and and it is present in near about rice growing all 85 countries. And wheat, we have also Coccidia graminis, which is also very devastating pathogen, which is also present on 365 cereals and grasses and present in all wheat producing countries. Recently, we have seen the example of UG99, that is Uganda 99 strain of Coccidia graminis triticide, which is has a capacity to have 100% crop loss, and that Uganda 99 strain of Coccidia graminis is now government of India also started building program against Uganda 99 case of Coccidia graminis. Then these fungals are very important, and they also causes losses in soybean rust, corn smut, wheat rust, rice. But these are the important fungal diseases which causes major loss and as far as the food production is concerned. Then as far as the plant pathogens are concerned, this plant disease, anything which prevents the plant from performing its maximum is maximum potential is called plant disease. Plant pathogens will restrict the activity of plants. The plant will not perform to yield, and the quality also reduces. And there are so many factors, like some biotic factors and some abiotic factors are responsible for plant diseases. We will see which abiotic factors are responsible for diseases. So many abiotic non-infectious diseases in which you can say nutritional deficiencies, then soil compaction, then salt injury, ice and sunscorch, zinc deficiency, you can see P and NACL stress in Jacopa, which will give you, there are so many examples of non-infectious diseases, or you can say abiotic diseases. And the important biotic factor which we build in plant pathology are all these living organisms. That's why we call it as biotic or infectious diseases, because it is contagious, infectious, and biotic living organisms. They are different pathogens. They infect tissues like leaves, shoots, stems, crowns, roots, tubers, fruits, seeds, vascular tissues, and this we call it as biotic infectious disease. Then what are the plant pathogens? Plant pathogens it's a broad term which includes microorganisms like your fungi, phytoplasma, viruses, and these nematodes, parasitic higher plants. These are all plant pathogens. They can make our ecosystem fragile, and then they will shake our food supply system. Plant pathogens also reduce the growth's ability to produce crops and can infect almost all type of plants. Every plant has plant pathogen. Only major plant pathogens are fungi. Different type of fungi, PGM pathogens. Then we have bacteria. It's a prokaryotic organism, fungi eukaryotic organism. Bacteria are single-celled organism, but having very, very devastating diseases. Then again, viruses are nucleoprotein, and viruses also they don't have their own cellular structure, but they have nucleic acid and protein, and with the help of this, they can use the plant cell for replication and multiplication. Then again, we have virals, which is only infectious nucleic acid, mostly RNAs, and this is also very infectious and causing losses to the plants. Then we have very tiny cytoplasma, which present in generally phloem of the plants, and which also reduces causes the diseases. And the nematodes are also nowadays we are finding lots of nematodes, lots of losses caused by nematode plants. Then, as far as stem parasites are concerned, we have Loranthus and Escuta goda, which classified as stem parasites, which grows on stem and which also disturb the physiology of plant and disturb the plants for giving their maximum potential. Then we have root parasites like Striga and Odobenchi, which is also causing some disturbances to plants. Then we have different type of symptoms. Symptomatology is a very different thing, that on the basis of this symptom and signs, we can identify whether the infection is caused by fungus or bacteria or virus or something like that. There are typical symptoms of fungus, like bird eye spots and berries, like we call it as anthracnose, Polypocratum fungus, which is responsible for anthracnose. Then damping out of seedlings, generally PTM and Phytophthora are responsible for damping out. Then lip spot, Septoria brown lip spot, then chlorosis, sometimes caused by fungi. Then the basic difference between fungal spot and bacterial spot is bacterial lip spot is always covered with yellow halo, and the spot is generally angular in shape, and that's why in cotton we call it the angular lip spot of cotton, which is called as Xanthonus bacterium. Then fruit rot, canker, and then crown gall, which is caused by Agrobacterium tumefaciens, which is a genetic bioengineer. We call it as a genetic bioengineer, Agrobacterium tumefaciens. Then viral disease, we have mosaic leaf pattern. It is a typical symptom of viruses: mosaic, crinkled leaves, yellow leaves, plant stunting. This is the major symptoms caused by fungi, bacteria, and viruses. Lip spot. You can see this lip spot caused by Cercospora on groundnut. Then this is the blight on maize caused by Phytoderma. Then wilt, generally wilt is caused by Fusarium. Then potato scab. The mosaic is a viral symptom. These galls, you can see galls in Agrobacterium tumefaciens. Grassy shoot, which is generally caused by phytoplasmas. These are the symptoms, various symptoms caused by various plant pathogens. Then, which are the different components of plant disease? How this disease grows? If you open the book of plant pathology, in every book, the first chapter is regarding disease triangle. Disease triangle has three components, like susceptible host, required in our field, in pathogen, which needs to be virulent pathogen, and favorable environmental conditions. These three factor composes the disease triangle. A favorable susceptible host along with the virulent pathogen, and which combines with the favorable environment, then there will be the incidence of disease in any conditions. Then these plant pathogens causes successful disease. There are different properties of plant pathogen. That is, virulent pathogen must have a level of virulence. Then that pathogen must be adapted to our locality, in our soil, in our area. Then it should have dispersal efficiency. Efficiently, it can be dispersed from one field to another field, from one plant to another plant. Then its survival efficiency, how it's going to be survival in a very harsh condition, and then its reproductive fitness, because this pathogen has a asexual as well as sexual as well as vegetative way of reproduction. Then these properties of host. Host are host means the crop which we are selecting in your crop, the which susceptibility. Suppose we have some susceptible cultivars, we have some resistance lines, we have some tolerance cultivar. If the host is susceptible, then and then only the disease will occur. If the host is resistant, then you can take the disease strangle. Therefore, susceptibility is very important. Then growth stages and form. On the different growth stages of host, their level of susceptibility is different. At the early stages, the plants are very vulnerable for infection of disease, and when it's progress, then they develop some pathogens or some phenol compounds inside plants, and which helps them to protect them from the pathogen. Population density and structure is very, very important. How much quantity of pathogen is available in the field, or soil-borne infection, airborne infection, seed-borne infection. What is the level of population density and structures will also important as far as the infection is concerned. Then general health of the host, which is also important for the infection of hosts. Then properties of environment, that is the triggering factor. If there is a host, susceptible host is there, virulent pathogen is there, but the environment is not congenial, then there will be no disease, because this temperature, rainfall, dew, deep wetness, spread, soil properties, and wind, this all will affect the this will cause act as a trigger for disease infection. Then, for example, if you take the example of potato lead blight development, in every disease there are different conditions of environment which are we call as a predisposing factors. Predisposing factor for the development of disease. These predisposing factors are different for different diseases. For instance, if you take the example of potato lead blight, the disease is more severe in cool and wet conditions. Then, depending on these conditions, we can predict the disease. When there are some cool nights, warm days, and extended wet conditions from rain and fog, then you can predict lead blight epidemics in this entire area. Therefore, if these conditions are available, then within 2 weeks the potato lead blight will be appear. Then there are various different chain of events which leads to disease development. And in disease development, the most important factor is, the virulent pathogen and the inoculation of the pathogen. That pathogen will land on the host, on leaf surface, or maybe on root, or where it infects. So that inoculation is important. After inoculation, it will penetrate and then cause infection. And then incubation is there. After incubation, it will reproduce, and then basically we will find out what is meant by inoculation. Arrival of pathogen on the host. That is very, very important. Then after it pathogen should penetrate, or it should invade in the host and by. It may be sometimes it will form like structure which will help to penetrate the penetration peg in, and these structures are different to the host cell. These are formed by fungi in organism. Then infection is successful establishment of pathogen with host tissues and derived nutrition from them. हा बोला.
थोडासा आवाज ब्रेक होतोय सर तुमचा.
हा बोला मॅडम.
सर तुमचा आवाज ब्रेक होतोय थोडासा. तर रेंज मध्ये आहात.
बरोबर, मी ते हेडफोन काढतो. आता येतोय का मॅडम आवाज?
कारण तुमचा आवाज थोडा.
आता येतोय का आवाज?
येतोय.
आता येतोय का मॅडम आवाज?
येतोय.
हा, आता मी हेडफोन काढून टाकलाय. हा, ओके मॅडम, ओके. Okay. So, as far as infection is concerned, the successful establishment of pathogen with the host cell or tissues, and when the pathogen derives its nutrition from the host, then we call it as infection. Then incubation, when the time interval between arrival of inoculum and the symptom development, that period between arrival and symptom development is called as an incubation period. Then reproduction, multiplication of the pathogen and its formation of secondary inoculum, it's called as reproduction. Then it's dissemination. Infected plant parts, wind, rain, insects, these nematodes, they are disseminated from one place to another place. So, for example, rust can disseminate from one continent to another continent. Therefore, the pathogen has different dissemination capacity. Then disease cycle. In every disease, there are two types of cycles. Some cycle we call it as a monocyclic or single cycle disease. The example is smut and rust, in which in monocyclic disease what happened, the primary inoculum, the primary infection goes and then it goes to overwintering stage. So only one cycle in that disease. The example is smut and rust. Then another disease cycle is polycyclic, or you can say multiple cycle disease. Multiple cycle disease, the example is lead blight of potato and then powdery mildews. In that we have primary source of inoculum, then primary infection, then again during the growth of that crop, there is secondary source of inoculum. This primary source of inoculation may be due to sexual spores, and this secondary source of inoculum may be due to asexual spore. In powdery mildew, you can say we have a Clistotricha ascospores as a sexual spore, and then Conidia as a asexual spore as a secondary infection. Then, which are the different components of plant disease management? That is the important topic which today we are seeing, integrated disease management. There are traditional principles of plant disease control, like we can control or manage the disease by avoiding, which is called as avoidance. Prevent the disease by selecting a time where there is no inoculum, that is we call as avoidance. We can avoid the primary source of inoculum or secondary source of inoculum to have a contact with host plant, that is called as avoidance. Then exclusion. Exclusion is the prevention of the introduction of inoculum. The perfect example of exclusion is nowadays we are hearing the buzzword like quarantine. Like also in plants also there is quarantine. We have domestic quarantine as well as international quarantine. If some inoculum or source of inoculum is coming from exotic, then restrict it, avoid it, so that you cannot have an entry of that pathogen in our area. That is called as exclusion. Then eradication, to eliminate, destroy, inactivate the inoculum, which we call as eradicant. We have different type of eradicant with us. Then protection, preventing of infection by means of toxicant like chemical toxicants or some other barriers of infection, that is called as protection. And then very important therapy principle is resistance, host resistance. In host resistance we have vertical resistance, horizontal resistance, so many resistance varieties we can develop, so that if there is a resistance, there will be breaking of disease triangle and there will be no infection. Therapy means actual cure of the plants. That means infection is already there, and because of that infection, we are applying some therapy or chemicals, therapeutic chemicals, so that you can cure that disease. These are the terms, but generally we cannot use the term plant disease control, but we need to think the term like plant disease management. We cannot eradicate anything from this earth. We can manage, we cannot control. That's why generally we use integrated disease management, not control. Then what is integrated disease management? It is the selection of different management control methods based on economic, ecological, sociological consequences. Then these ideal practices remove or alter the condition that attract or conducive for pathogen infestation, and therefore IDM can better cure existing infestation and prevent future one. This is the cumulative effect. Whatever the management tactics is available with us, that we have to use judiciously. These are the various different methods which generally we use in integrated disease management. Generally, we start with agronomical, that is cultural practices. Cultural practices are very important. If we use this tactically, we can manipulate, we can avoid the disease incidence in various crops. Then nowadays, very important and gaining very much importance as far as the organic farming is concerned. Then the effects of chemicals after the book of Silent Spring, there is a biological control of management which is increasing very fast. We have so many kinds of biological pesticides are available, and so much research is going on, and biological method is getting popularity to avoid the diseases. Then host resistance is always there. It's the conventional methods. Host resistance is a continuous process to breed different resistant varieties for different crops. That is the very efficient and very economical way of management, eco-friendly way of management of diseases. And then another physical methods are also very important, like you can use some solar treatment, you can use some hot water treatments. So many physical methods like mulchings are also now using so many farmers for avoiding some soil-borne plant pathogens. Then the last option is with some chemicals, having some green chemistry. Nowadays, very good chemicals are also available, which is having less residue level and having derived from some biological origins, like strobilurin group of chemical fungicide is derived from mushroom. These kind of chemicals we need to use, and there are so many other things are also available for integrated disease management. Why to adopt IDM practices? Because we can see that there are hazardous effects on humans, animals. There are in Punjab, in Kerala, we are seeing some cancer express are running because of the high, excessive use of pesticides and chemicals. There are residue levels. Our products are not accepted worldwide. Our products are rejected due to the high incidence of residue. Our grapes are consignments are rejecting. So many things are there. That's why we are very promoting the integrated disease management practices for management of plant diseases. The principle of IDMs are it's allows some tolerable disease development. Then it's because there are some friendly organisms are available in the field, and that will take care of this tolerable disease which already developed. Then we can utilize natural control measures so that it will be eco-friendly. It will has less impact, less damage to our ecosystem, less damage to our environment. That's why maximum application is utilization of natural control measures. Then consider the environmental and the natural ecosystem. That's obvious for disease management. Then this simultaneous we can manage multiple pathogens with the help of integrated disease management. Then we have regular monitoring of pathogens so that we can have a preventive measures, so that because most of the methods are if we protect preventively, then we can avoid the damage of pathogen, the damage of crop from the severe infection. Then use at economic threshold level, that is very important. That's why you have to have some monitoring. Always you monitor which disease is growing, where is the area where disease is growing, and then you can act this, then utilize this idea judiciously. Then what is the advantages of integrated disease management? The advantages of integrated management are we have better knowledge to develop this idiom. You should have some knowledge with you. You can utilize less toxic, more targeted techniques. It is more effective, then cost effective, cost is less, then risk is less, and each require collective efforts from the farmers' point of view. Now, these are the various options available. These are the options available with this integrated disease management. We have cultural methods like site selection and sanitation, crop rotation, which is very useful, which will alter the pathogen life cycle and which will change the life cycle of pathogen. That's why this cultural method will help to reduce the disease. In physical method, we have pruning, mulching, barriers, weeding. These weeds are also act as a lateral alternate host, which if we remove that, we will break the life cycle of pathogen. And if you break the life cycle of pathogen, then it will not develop this secondary inoculum. That's why physical method is also very important. And you know, in biological methods, antagonism is very important. In with we have a very good example of trichoderma, which has a mycoparasitic ability on some soil-borne fungus like PTM phytophthora. Then it also have a hypervirulence, that is some strains of that same pathogen will also kill the similar pathogen. And antagonism, we have antibiosis, which secrete some antibiotics, which will inhibit the growth of plant pathogen fungi. And chemically, we have so many insecticide, fungicides, and herbicides are available in market to manage these fungus or these diseases. In this, we have this prevention activity, then avoidance, and then monitoring and suppression, which includes this. There are various tactics of reducing the initial inoculum. What is meant by management? Management means reducing inoculum level, initial inoculum level. This inoculum level we can reduce by avoiding the primary source of inoculum, then exclusion, we know, by with the help of quarantine, eradication, then protection, resistance. These are the way of reduction of initial inoculum. Then you can also reduce the infection rate. If you reduce the infection rate by with the same principle like avoiding, exclusion, eradication, protection, and resistance, we can also manage the disease. Then the epidemic is also very popular word nowadays. Pandemic is very popular as far as corona is concerned. But in case of plant disease also, there is the epidemic situation is always there. Therefore, you have to reduce the duration of epidemic of the pathogen. Then we will quickly see what are the different emerging plant pathogens worldwide. Very quickly we will see because of this is climate change and geographical distribution. This is the phytophthora causing various diseases and very devastating. And this is Irvinian very aggressive. It's caused by Hemiplegia, which is also emerging disease. This is phytophthora in South India is also now emerging as a big blow vegetables. And this phytophthora infestans. Then this dry root of chickpea, Rhizoctonia bataticola. This is also emerging as a very devastating pathogen as far as chickpea production is concerned. Then we have different cultural practices. It is one component of integrated disease management. And in cultural practices, we can reduce the disease. And these cultural practices, deep plowing is also very important. And there are some pre-planting operations before plantation of plant. We can choose choice of site where primary inoculum is less, clearing of land, strumbling, plowing, deep plowing, harrowing, ridging. These are the operations which will certainly help to reduce the inoculum, primary source of inoculum. Then these are the activities done during planting. You can do transplanting, nursery practices. That will also help to reduce the inoculation, primary source of inoculum. Then post-planting operation, you can thin the plants. Because of thinning, there will be less microclimate and less fungus will grow. Then supplying mulching. These are the various operations which will also help in cultural methods the important cultural practice is rotation with unrelated crop. Why we always recommend you rotate your crop? Because after one crop, if one crop will grow, the pathogens of that crop will grow, and they will also be available in the field for the next crop. If you rotate the crop with another crop, then there is a host specificity in pathogen. The good thing is host specificity. They will not cross infect to other plant. Therefore, because of that host specificity, if we use the rotation, if you rotate the plant, then you will reduce the damage from the disease. That is very, very important. Then fertilizer uses also, some fungi are nitrogen loving. Some, if you give more nitrogen, there are more effect of fungi. Some fungi are potash loving, so that you can also judiciously use the fertilizer so that you can manage the disease. And if you cultivate deep plowing and deep intercultural operation, you will put your inoculum to the soil depth which is not accessible for the root of that crop. Therefore, you can use this for cultural management. Then if you use raised bed technique, or nowadays we are also recommending this technology for various crop, and the results, the reason behind is that the primary source of inoculum, the source of inoculum will not be accessible to the roots of the plants. Therefore, you can plant on the raised bed system. That is also help. Then if there is an infection, severe infection, there are overwintering stages of pathogen, and if you collect and destroy or burn that overwintering stages, you can reduce the primary source of inoculum also. Then time of sowing, you can escape the infection of pathogen. Suppose in case of wheat, if you delay planting of wheat, which will escape the chances of wheat streak mosaic virus. Then in early spring planting of cotton may effectively help escape of cotton root rot. That is a escaping phenomenon. Based on that, you can adjust your date of sowing of your crop, which will also help you to reduce the disease. Then removal of undesirable plants, because some plants are act as a overwintering or alternate host for pathogen. If you remove such undesirable plants, you will break the disease life cycle of that pathogen, and if the life cycle is disturbed, then there will be no multiplication of that pathogen in your field. Likewise, in the wheat rust, we have some barberry as an alternate host. If you remove the barberry plant, like undesirable plant, which will break the disease cycle or life cycle of Puccinia graminis triticide, and which will help to manage the rust in wheat also. Then these are the some plants which help for infection, which you can remove. Always we recommend roguing removal of disease plant from most of the viruses. We generally advocate these roguing practices, or in seed production product also we rogue the plants which we don't want. And viruses disease and bacterial wilt disease of cucurbits are the example where roguing is very, very important. Then the another most important strategy of integrated disease management is host resistance. Host resistance is, there is constitutive resistance and inducible resistance. Like this is a plant defense system. In plant defense system, there are some constituted, or you can say active defense system, or some inducible or active, or constituted or passive. In this, we have a structural and biochemical resistance mechanism. In structural, you can see the plant has a wax layer, cuticle thickness, cell wall, stomata, shape and activity, and lenticel. These are the structural constituted way of plant defense. And also some biochemical, like preformed inhibitors, example phenolics and hydrolytic enzymes. Some phenolic compounds are present inside the plants, which will help plants to have a resistance against some pathogens. And some inducible, that means some if you apply something, this resistance will induce. that is called as inducible. In that, because plants also have its defense system. When plants see that there is an infection of some pathogen, it will form cork layer, it will also form abscission layer, also develop some gums, then also papillae and tylosis. This is the structural defenses by plants. And some biochemical like hypersensitivity. Then phytoalexins are the chemicals which are developed in response of infection. Then SCR, that is systemic acquired resistance. They're like salicylic acid we spray, then the plants will acquire this systemic acquired resistance. And these antimicrobial enzymes are also produced after the infection of plant pathogen. Then pathogens that keep their host alive and feed on living plant tissues are called as biotroph. And the biotrophic fungi are the example as albugo candida. This is the albugo candida. This is biotroph. Then we have some necrotroph, which has ability to degrade the enzyme and have necrotic patches on leaves. Example is Irvinia disease or botrytis. This is a necrotroph. Then we have some hemibiotrophs. Some pathogens are biotrophic. Early stages they are biotrophic, and after infection they become necrotrophic. Therefore, they are called as hemibiotrophs. The Colostotrichum fungi is a very good example of this hemibiotroph. Then this pathogen have a compatible response as well as incompatible response. Then there is some immunity and some higher susceptibility. These are the some basal resistance as well as then the plant immunity. And sometimes what happens when there is an infection of pathogen, then the plant will kill the cell it's around. That is called as hypersensitive response or HR response. Plants may respond to the situation with another defense because it will deliberately kill the plant cell around the site of infection. That is called as hypersensitive response. This is also a defense system of one by plant. And there are some structural defenses because the plant cell wall is made up of cellulose, then cross-linking glycans, this hemicellulose, these fibers that will help to avoid the infection from fungus and will protect from fungus. These are the structural defenses. Then many cell wall also have lignin. Lignin also is it is a heterogeneous polymer composed of phenolic compounds. Phenolic compounds always used in defense system in plant that gives the cell rigidity. Lignin is primary component of wood and cell wall that becomes lignified and will help. Then cutin, suberin, and waxes are fatty substances that may be deposited in either primary or secondary cell wall and further protective tissues for the plant, and which will help in inducing the defense mechanism. Then we have also callose layer, then papillae, which will also in response with the infection of pathogen that will develop, and they will help to go further infection of plant pathogen. Then some chemical also secondary metabolites are produced by plants and which will also help to avoid the infection. Then some proteins and enzymes, these are also produced in a systematic acquired resistance. When we apply some salicylic acid, these chemicals are developed inside the plants and it will help as a induced resistance. Then we have two types of resistance. One is vertical resistance and another one is horizontal resistance. A plant variety which is resistant again only one with the only one gene, that we call as a vertical resistance. And horizontal resistance means it is governed by many genes and which is with the more pathogens, it is susceptible for more pathogens, and therefore we generally recommend horizontal resistance which will last longer than vertical resistance. Then another tactics which is very important in integrated pest management and nowadays it is very popular is called as biological management. Is in biological control method, this trichoderma and this so many fungi are available which are called as biocontrol agents. And this biocontrol agents, the way of this biocontrol agents is antagonism. That means mycoparasitism, antibiotics, and competition. This is the main part of antagonism. Then induced systemic resistance or SAR, systemic acquired resistance, when we apply this trichoderma, this biocontrol agent, the plant induces its resistance system, and then it will protect from the pathogen. Then competition, when we apply these agents, biocontrol agents, the population of biocontrol agents will be more in compare with the plant pathogenic fungi like Pythium, Phytophthora, and because of that competition, the pathogenic fungi will not be able to infect the plants. Then this hyperparasitism, or you can say antagonism, this Trichoderma hergianum hyperparasitizes with the rhizoctonism. You can see here the Trichoderma fungal hyphae will coil the hyphae of rhizoctonia and will kill that rhizoctonia and take nutrients from that rhizoctonia. That is called as mycoparasitism or hyperparasitism. That is the one way in which this biological control works. Then this Anthracotrophis obliqua develop a net-like structure for trapping the nematode. This is also my example of mycoparasitism. Then you can say if you put Trichoderma or any bacteria in a Petri plate along with the plant pathogenic fungi, you will find antagonism there. The Trichoderma will produce some antibiotics or some lytic enzymes, some unregulated product which will inhibit the growth of pathogen. Then competition is indirect antagonism. When we put more amount of biocontrol agent in the rhizosphere area, there are plant pathogen as well as biocontrol agent. If you put more biocontrol agent, there will be competition and the plant pathogenic fungi will not survive. Then there are the latest paper also has a reference that this Trichoderma fungi, when we apply, which induce the host resistance, like Pseudomonas fluorescens also has a capacity. When we spray or when we apply in the soil, the plants will triggered for its host resistance. Their jasmonic acid pathway, their salicylic acid pathway will be triggered, and this systemic resistance will induce and will help to increase its immunity of the plant. Then these are the suppressions, and now the very important method which in integrated disease management we generally advocated, use last option as a chemical management, because we have so many hazard effects, but nowadays we have very good chemicals are also available. These chemicals are of different types. We have some eradicants, some protectants, some therapeutical chemicals. These are the various classification. The chemicals which kills bacteria, we call it bactericides. The chemicals which kill nematode, we have nematicides, then fungicides. There are so many chemicals are available in the market, and these also they are categorized by protective as well as curative. Because if we have a possibility of there are some predisposing factors and we are looking for there may be infection of any fungi, then we can go for a protective way of protection. And then if there is already infection of any pathogen, then you can go for a curative way of pathogen, a curative way of protection. And this is the different group, like some are absorbed, some are translocate, some prevents infection or sporulation, some use before infection, like strobilurin that I have already mentioned. That strobilurin is a group which is derived from mushroom, and chlorothalonil. These are the some protective fungicides. And in curative also, some absorb, some translocated, some kills fungal tissues, use after infection. And the example is all these difenoconazole, tebuconazole, propiconazole, hexaconazole. All these triazole groups comes under this curative fungicides. Then eradicants, seeds, bulbs, corms. You can also eradicate if you are exporting some fruit to the importing country are asking eradication facility. They are also useful for killing of some infection from the plants. Then these are the some protectant protective spray we can use, protective dust also you can use, and this will protect your crop from getting entry of pathogen inside the host tissue. Therefore, these protectants you can also use for spraying. Then there are some chemicals which act systemically. That is called as systemic fungicides. This systemic fungicides can absorb by plant without harming it and transported to other tissues where they are toxic to fungi. When the fungi, there are some intercellular fungi and some intracellular fungi. The fungi when are going inside the tissues of the plant, then you need systemic fungicide. When you use contact fungicide, the fungicide will not reach to that fungus, and therefore it is not able to eliminate that fungus with the help of contact fungicide. In that case, you need to use some systemic fungicide. These compounds can control and eradicate established infection. Already infections are established, and then it will go reach there and will cure the fungus. But they are also vulnerable to development of resistance, because when you repeatedly use a systemic fungicide one after another, then there will be chances of getting resistance in that pathogen against that systemic fungicide. Therefore, you have to use different systemic fungicide at different time. Do not use one systemic fungicide regularly. Therefore, to avoid this resistance, you have to use different group of systemic fungicides. Then some antibiotics are also very important, but also antibiotics also has a possibility of getting resistance against bacteria when we use continuously. There are some streptocycline sulfate, streptomycin, and cyclohexamine. These are some example of antibiotics. Copper oxychloride is also acting as a sometimes antibacterial agent. Then nematicides are also very expensive, and that's why they should be used very judiciously. Then after this, we have these options like chemical, biological methods are available. But the important thing is how the delivery system. What is the method of application? Method of application is very important. In soil fumigation is also very important method. In most of the vegetable crop, in most of the nursery area, where nurseryman generally prepare a potting mixture, and this potting mixture needs to be disease pathogen free. If there is any spore of pathogen Phytophthora, then there will be disease will be occur in that plants. Therefore, soil fumigation you can apply with the solar plastic mulching also. If you give water first of all, then mulch it and then apply with the soil fumigation. Then you can also have a soil drenching, because so many wilting, so many fusarium, so many ceratocystis wilt, so many wilts problem are in many crops, like in vegetables, in fruit crops, and crops. In that cases, you need to drench the soil with the chemicals, so that it will avoid the contact between your host plant and pathogen, and it will kill the pathogen. This is another way of application. Then nowadays you will find so many crops are irrigated with the drip system, and with that drip system you can also use fungicide, apply fungicide with the drip water, so that it will reach to the rhizosphere area and it will manage most of the soil related diseases or soil borne plant pathogens. Then there are so many seed-borne plant pathogens. There are internally seed-borne pathogens. There are externally seed-borne pathogens. Like loose mud of wheat is an internally seed-borne pathogen. The pathogen lies between the epidermis. And there are so many viruses also present inside the seed. That's why seed treatment is the very cheapest method of seed disease control. You can treat the seed with some seed tracers, some Captum, Thyrum, so many Vitavax. These are the chemicals are generally used for as seed tracers. And nowadays we can use Trichoderma, Pseudomonas. These are also biocontrol agents, and we also can use as a seed treatment to avoid the soil-borne plant pathogen. Then there are sometimes there are foliar diseases. Foliar application our spray technique is there should be some chemicals we need to spray with the foliar application, because of there are some aerial transmission of fungi, the aerial dissemination of secondary spores, which will disseminate from one plant to another plant. And to avoid that secondary spread of disease, we need to spray the plant with some chemicals. In that, we have so many sprayers are available with us, and with that spray we can use the spraying with the chemicals. Then also in nursery we need some transplanting, and during this transplantation, if you use chemicals, in that chemicals you dip the plants and then transplant. It will help you to avoid the seed-borne infection. Generally, carbendazim and chlorpyrifos is used for this method, because to avoid some pest as well as some soil-borne plant pathogens. Then seed bed also you can treat with the dazomate granules. So many chemicals are available to avoid the oospore infection or the soil-borne oospores which are available in the soil, which causes damping of diseases like pythium, phytophthora. To avoid this, you can use seed bed treatment. Then after this, because we have RAVE student with us, and RAVE student have maintaining their manual of disease. The why disease is very important, and in their manual they need to measure the disease. We will see the why disease measurement is important, because to know the prevalence and extent of the damage caused by disease and to develop the effective management strategies, we know how much disease is there. Therefore, they will measure the disease. And this disease damage is sometimes whole plant is killed or damaged, or sometimes there is a localized part, localized plant is infected. The effect of disease outbreak persists over several seasons. These are the various possibilities of diseases. Then disease is measured in terms of intensity, and disease intensity can be expressed as disease incidence or disease severity. The RAVE student must have gone through this, because they have also already calculated, I think, this disease incidence and disease severity from their host farmers field. The choice between evaluation of disease according to the incidence or severity depends largely on type of disease and its objective. There are two ways of disease intensity expression, which is disease incidence. One is disease incidence and another one is disease severity. The disease incidence and disease severity, what do we mean by disease incidence? The disease incidence means percentage of disease plant or parts in the sample or population of plants. It can be the proportion or percentage of disease leaves in a plant, disease stalks or tiller or disease seedlings in a field. If you want to calculate disease incidence, then you can calculate number of infected plants and divided by total number of plant assays. Suppose in a row you have a 40 plants, and in that 40 plants you have a number of infected plants 15. That means number of infected plants will be 15, and total number of plants which we have assessed, maybe in a row you have 440. You can 50 divided by 40 into 100. That will give you disease incidence. How much disease incidence is there, that you can calculate. Then disease evaluation according to the incidence is suitable for most diseases in early stages of their epidemic, and it applies mainly to diseases which affects whole plants, such as systemic viruses, disease, weeds, mers, fruits. In that cases, you can use disease incidence. Then disease incidence generally tells about the prevalence of the disease in a given area or host plant. If you found out that in a given area how much disease incidence is there, then you can calculate that way. Then another way of expression of disease is disease severity. Disease severity is a percentage of relevant host tissues or organ covered by symptoms of lesion or damage by disease. Severity results from the number and size of the lesions. Then disease severity is more appropriate in disease like rust, downy mildews, powdery mildews, dip spots, and other similar diseases. Disease severity tells about the extent of damage caused by the disease. And then how you will calculate the disease severity, or sometimes it is also called as infection index. In this, you have to use disease rating scales. Disease rating scales are there. In that scale, sum of all disease rating divided by total number of rating into maximum disease grade into 100. We will see like this. There are so many standard area diagram, that is some disease cycles are there, disease scales are there, like these are standard area diagrams are there for leprous, for so many diseases, 1% infection, 5% infection, 10% infection, 20% infection, 50% infection. Likewise this is the standard diagram. One is used for leprous, another one is the stain rust. Here is 1% means this much infection, 5% means this much, 15% means this much infection, 25% means this much infection. Likewise, these are the 1, 5, 25, 50% infection. These are the disease rating scales. Likewise, this is also for gloom blotch disease, and these scales are very important to find out disease severity. How you will calculate disease severity? Suppose this is the scale in which there is no disease, that means 0 scale, no disease. If there is 1 rating, that means small brown spot covering less than 1%, and this 3 brown sunken spot 1 to 10%, 5 means brown spot 11 to 25%, 7 means circular 26 to 50%, and 9 means circular more than 51%. Then this scale, how you can use to calculate disease severity? Suppose this is scale 0, this is scale 1, this is scale 3, then 5, then 7 and 9. This is the scale which we are using for calculating disease severity. And what you will do, you will grade it. Your grade, suppose your grade is 5, total rating is 5. Then in second you have 5. Then in next you have 4. You have 8. Likewise, total rating which we have get is 34, and number of ratings which you got is 186. Then sum of all rating is 186. Total ratings are 34, and maximum disease grade is 9. If you want to calculate disease severity, that means 186, which is your sum of all rating, which is 186, and which is divided by total ratings. Total ratings you have done is 34, and the maximum disease score is 9. Then 186 divided by 34 into 9, and all this figure is multiplied by 100, that is 60%. That means your disease severity is 60%. This way you can calculate the disease severity. Then after that, we can have a safe and effective handling of agriculture chemicals, because after this Yavatmal incidents, there are so many risks is there. You know, the pesticides, we know different type of pesticides are there which we use for management of diseases. Then insecticides, fungicides, herbicides, nematicides. This should be, there are different groups. This should be used, the similar group of pesticide should not be used at the same time, because to avoid the resistance. Because if you use the same chemical from the same group, then it will, then there are possibility of development of resistance against that chemical. Then we have contact pesticide, systemic pesticide, translaminar. This is some are effective on respiratory system, some are so many mode of action is there. Then this is the precaution you should take while always using these chemicals. Then there should be some pesticide mixing sequence. these are the formulation. First of all, according to this, you have to mix this pesticides. Then while applying these, the spray solution pH should be not more than 8 and not less than 5, and then the EC should be not 1.5, and do not apply hot sunny days. These are some precautions you should take while applying the pesticides. And this your nozzle size is very important. 250 micron is very important as far as the pesticide is concerned, not more than this. And you nowadays we have water which is having more pH and more TDS, and that will affect the spray solution for alkaline hydrolysis, and which will result in different chemical reaction which we don't want. Therefore, always have a pH meter with you and test your pH. The pH around 6.5 to 7.5 pH is required, and the TDS is around 100 only, because most of the water is having more TDS, like 4,000 TDS may be there, and that will hamper your EC. Then there are incompatible mixtures. We cannot mix some chemicals with the another chemical. This is the list is there. Borders mixture and this, all these things are there. Then this pH also affect the pesticide. There is alkaline hydrolysis, and which there will be reversible and irreversible reaction. Because of the pH more than 7.5, around 8, there will be different kind of reaction, and you will not get the result from the fungicide or the insecticide because of the pH of pesticide. Then hardness of water, TDS should not be more than 100. Generally, preparation of solution is very important. What do you mean by 10 ppm, 20 ppm, 250 ppm, 300 ppm? If you want 1 liter solution of 10 ppm, then you have to use 0.01 gram per ml. That is called as 10 ppm. If you put 1 gram in 100 liter water, that is 10 ppm. If you put 2 gram in 100 liter of water, that is 20 ppm. If you put 25 gram in 100 liter of water, that is 250 ppm. 100 liter of water, that is 300 ppm. If you put 50 gram in 100 liter water, that will calculate. How you calculate this? Then sometimes there are recommendations that use 0.1% spray. What do we mean by 0.1% spray? 0.1% spray means 1 ml in 1 liter of water. That is called 0.1%. What do we mean by 0.2% spray? That means 2.5 ml in per liter of water. Likewise, we have different calculation for 0.15%. 0.5% means 5 ml per liter, 10 ml per liter 1%, 5% means 50 ml per liter means 5%. These are the different calculations for preparing solutions. This is the formula for preparing. Suppose you want to prepare 10 liters of 500 ppm, then you have to buy ppm, that is 500, quantity of solution 10 liters and 10,000 liters divided by 10 lakh. This is the nowadays the government has banned these chemicals. Already draft is there, and government is planning to ban these chemicals which are available in market. And in a coming future, we need to find out other alternatives for these chemicals, because this in future this chemical like carbendazim, mancozeb, then monocrotophos, this will not be able to use in our farming system. Therefore, this is the draft, and already these are banned in European Union, and in the future in India also it will be banned. I think this is regarding my integrated disease management. Thank you very much for organizer for giving me opportunity to present my lecture in front of you. Thank you very much.
Thank you, sir. So here, good afternoon, everyone. I am very grateful to Professor Mule ma'am to give me opportunity to give the vote of thanks to everyone. Here I am very much thankful to Dr.G.p. Jagtap sir. He give very such grateful information to our Rave students, and he will definitely convey this beautiful information regarding the integrated disease management to their post farmer also. Sir, give the detailed information related to the integrated disease management. That is how you should control or manage the disease over the various plant with the help of different kind of the management methods. That is agrochemical, biological, physical, then chemical, host resistance, and other also. So you should don't use only the one method. You should use all these combined method to control over the particular diseases. So it will be the helpful to our ecosystem, to protect our ecosystem, and also enhance the harmful effect regarding the plant diseases, animals, and human also. Sir, very much thankful to you from College of Agriculture, Kanchanwadi, from our Agriculture Extension Department. I also thankful to our Director and Principal, Dr. D.K. sir, and also all attendees who are participating in this webinar, and they carefully listen all the information said by the Dr. G.p. Jagtap sir. And also thankful to all the professors in our college. So, sir, very much thankful from Agriculture Extension Department, College of Agriculture, Kanchanwadi. Thank you, sir.
Thank you. Thank you. Thank you, madam.
Thank you, sir. Okay. Okay, all can leave the meeting. Our webinar is over. Thank you very much.